Good evening and good morning to everybody. Welcome to the third class of this series. And again, I want to congratulate Professor Liao and Professor Joaquin for putting together this, this incredible experience of teaching online, which is seems it's going to be the future. The aspect that I want to discuss with you this morning is associated with the role of inflammation during the process of implantation. And I'm going to discuss and show you a little of data how inflammation is an evolutionary process necessary for the success of pregnancy. Activate this. Let's see. And I need, yeah, okay. So Professor Joaquin made my life much easier because he already introduced you yesterday everything that is about the implantation process. So I will not go in much detail, just to remind you that the most important stages in the process, in the early process of fertilization is happening in the fallopian tube. This is in humans, of course, in the fallopian tube, in the ampulla, where is the meeting between the sperm and the oocyte. Then the fecundate oocyte, the blastocysts start dividing into the 2 cells, the 4 cells, 8 cells, the morula, and then it reaches what I call this the Grand Canyon, that is the uterus. An incredible and huge a cavity that, in principle, as I will discuss a little later, is against implantation. So, contrary to what we think that the uterus is always prepared for the implantation, is the opposite. The uterus is not ready to implantation. Only in a specific area and in a specific time during the menstrual cycle that we call the window of implantation. And again, in a specific location at the wall of the endometrium, the blastocysts will implant. And this is when the pregnancy will take place. Again, this time, this window of implantation was defined many years ago. Is still, some people, it is usually thought between the 8 and 9 after fecundation, or between the day 19 to 21 of the menstrual cycle, is a subject of debate. Is this window of implantation happening in some women early or later? Still, we don't know. But in general, we define the window of implantation, that small time where there is a place in the endometrium that has undergone changes that will accept the embryo. The implantation process is defined in at least, we'll talk there is more, stages. One stage is the apposition, where the inner cell mass now is looking towards the epithelium. There is the time of attachment, that is the physical interaction between the trophoectoderm, external layer of the blastocyst, with the surface epithelium of the uterus. And the third one is the invasion, when the trophoblasts start migrating out of the blastocyst and penetrating into the endometrium. So if we look now at this picture, I was mentioning to you that the uterus is not prepared for implantation. And why? Because the uterus, especially the epithelium, the surface epithelium of the lumen, is a mucosal immunity, is a mucosal tissue. Similar what you see in the lungs, similar you see in the gastrointestinal tract, or even in the skin. Any mucosal tissue is covered by layers of mucins. It's the mucus. And the role of the layers of mucins is to prevent anything to attach and penetrate. It's the first physical, if you want to call, barrier against the invasion of pathogens. So we know that bacteria is present in the lumen of the uterus. So the uterus has developed this capacity to protect against the penetration of bacteria. We will talk in the next lecture when we talk about microbiome and infections in the pregnancy, how these mucins are very important in the protection against infections. So the mucin is covering the whole surface epithelium. So when really the blastocyst is reaching the uterus, it sees a surface that it cannot bind. In order to have the apposition and the attachment, this mucin needs to be removed. Then the epithelium needs to express adhesion molecules, who are the hands that the mother, the epithelium of the mother is extending into the baby. And once those two hands are one with each other, when there is the connection, the physical connection between the adhesion molecules in the epithelium and the adhesion molecules in the mother, then the attachment happens. But that just is a first stage. It's not even a stage that will confirm the pregnancy. The real pregnancy, or at least the early stage of the pregnancy, what we call the real stage of implantation, is only going to happen when the trophoectoderm start migrating, becoming trophoblast cells. And this migration goes between the epithelial cells and between the epithelial cells and the surface. And then they will start now, so a phase of migration, and then we start the process of invasion. And there is an early invasion that it will go through the stroma cells in the uterus. When they reach the stroma cells, when this invasion happens, now the embryo is seated within the maternal, within the endometrium, and at that time we can call it an implantation has occurred. So what is this important? Dr. Joaquin mentioned also a little bit about this yesterday, because a failure of implantation is extremely relevant, especially related to recurrent pregnancy loss. And yesterday was discussed about that, so I'm not going to talk about that much, just to remind you that 15% of all the couples are as affected by infertility. So the capacity of the endometrium to receive the embryo is the limiting factor, is the limiting step during implantation and the pregnancy success, especially in those patients who have normal embryos, anatomically normal endometriums, hormonal they are okay. The problem is the implantation. Therefore, the importance of understanding the basic signs associated with the process of implantation. If we don't understand what is the biology, the cellular and the molecular factors controlling the process of implantation, we will not be able to help those patients who are failing to conceive, or at least to implant. So again, many factors has been associated with the infertility. What we are focusing is the aspect of immunology. This is a reproductive immunology course, so whatever is immunology is what we're touching. And as I mentioned to you before, for many years it was thought that the problem, the immunological problem at the implantation site or on the time of implantation was due to a rejection of the embryo, the classical Medawar hypothesis that we discussed at the beginning. But now we know that the pregnancy is a more complicated thing, a more complex, let's put it in that way. It's a more complex process. It's not just a simple graft versus host response or simple rejection, because we talk about that there is a lot of immune cells present there. And I mentioned to you, just to remind you, that those cells are extremely important for the whole pregnancy. So what we want to understand is which one of those immune cells are necessary for the early stages of implantation, for the early stages of preparation of the uterus on implantation. And you heard yesterday a lot about the NK cells. I am not going to touch the role of NK cells today. The aspect that I mentioned to you before is that pregnancy is divided in these three immunological stages. A first trimester that is inflammation, a second trimester that is a Th2 anti-inflammatory condition, and a third trimester that is inflammation again. So the question for today is, why do we need inflammation in the early implantation and the early pregnancy, at the time of implantation and early presentation? What is the role of inflammation in those early stages? So the first thing we do is, I show you here, what are the players? So you have a player that is the blastocyst. A second player is the epithelium. Both of them need to be prepared to interact. So that is stage one. The second stage is, if they interact, now the trophoblast has to invade. And when the trophoblast invade, you need new blood vessels, you need tissue repair, you need now to start forming the placenta. So we can divide the inflammatory process of the role in the immune system in those two aspects. And that's what I'm going to discuss and dissect with you. A first stage is this, what it calls the apposition and the attachment, the preparation of the epithelium. And the second stage is the migration and the invasion. What are the immune cells who are playing a role in this today? So let's start with the preparation of the epithelium. As I mentioned to you, the epithelium is covered with this mucin, and as you see in the graphic, if there is a mucin, the blastocyst cannot attach. So the factors coming from the immune cells need to remove that mucin, number one. Second, the factors coming from the endometrium needs to induce the expression of the adhesion molecules. And when that has happened, when we have removed the mucin and when the adhesion molecules are expressed, then and only then the blastocyst will attach. And the first part of the implantation process will occur. Only the first part. So the questions that we have been bringing is, what are the factors that remove the mucin layer covering the epithelium, and also what regulates the expression of the adhesion molecules? So interesting, because the whole story started with a clinical observation by one of my collaborators, Dr. Nava Deckel, when she observed that a biopsy of the endometrium was inducing an inflammatory response that facilitated the preparation of the endometrium for implantation. So what was the study and what I refer to the biopsy? So what her team did is that they wanted to see what is the status of the endometrium. So they will go inside of the uterus, they will take a biopsy of the endometrium to analyze, and then in the next cycle of the in vitro fertilization, they will transfer the embryo. And to their surprise, those patients who had a biopsy of the endometrium, they have double the rates of implantation, clinical pregnancy, and live birth. So something was happening by just making a little wound in the endometrium that facilitated the implantation of the embryo in those patients. So we collaborate and we start looking at what are the changes that are happening in the endometrium. The biopsy was taken in the day 8 of the menstrual cycle, and then we take another biopsy at day 19 of the 21 of the menstrual cycle. So close to the end of the window of implantation, to see if the first biopsy has done something to the tissues. And we had as a control a biopsy that was taken only in the day 21st. So the endometrium was not exposed to a previous wound. We analyzed using flow cytometry for the presence of immune cells, and to our surprise, the only cells that we saw that were increased in the samples who had a previous biopsy were cells that were labeled as HLA-DR positive, CD11C positive. Again, this is human markers. These markers correspond to dendritic cells. And the interesting thing is, you can see here, this is the biopsy in day 8 or 12, has a very low number of dendritic cells of HLA-DR positive, CD11C positive. However, when we take the second biopsy in day 21, in all those patients, we see that the number or the percentage of HLA-DR CD11C positive had increased significantly. In day 21, of patients who did not have a previous biopsies, HLA-DR CD11 positive were very low. So their wound was able to reactivate or to do to trigger a response that was recruiting a lot of dendritic cells, who will still be able to see it 15 days after the biopsy, or 10 days after the biopsy was taken. So we further analyze what are the cytokines and growth factors that were produced within that endometrium 8 to 10 days after, so 10 to 12 days after that the biopsy was done. And I'll give you also additional information because of time. The arrows are showing you those patients who later got pregnant. So you can see that if there was a first biopsy, in the second biopsy, with the characteristics of those tissues is the expression of high levels of pro-inflammatory cytokines, including growth alpha, IL-15. You heard yesterday about IL-15 and NK cells, MIP-1 beta, and also the messenger. This is the protein concentration, the RNA, increase in MIP-1 beta RNA, and increase in TNF alpha, and another ones that I will discuss a little later. No one of those cytokines were changed when we took the first biopsy in patients, even at day 21, who never had a biopsy before. So clearly, that wound was able to induce an inflammatory process. So with that human information, we went to the mouse, and we look what happened if in the mouse we deplete the dendritic cells. Yeah? So here I'm showing you the early implantation of the mouse embryo in the uterus. This is the mouse uterus, this is the embryo, and it has implanted. If we remove the dendritic cells from day 0.5, so immediately after there is the plaque, and we look what happened at the time of implantation, you can see here. The embryo is present in the uterus. So you see clearly the embryo here, but it's not able to implant. It tries to establish a relationship to bind to the uterus, but as you see here, it fails that and it will fall down. So if the dendritic cells are not present at the time of implantation, the embryo is not able, the blastocyst is not able to attach firmly to the surface epithelium of the uterus, and the pregnancy will fail. There will not be implantation, and of course, there will not be a pregnancy. So we ask ourselves, are the dendritic cells or the factors produced by the dendritic cells preparing the endometrium for the receptivity of the blastocyst? So what we did is we took epithelial cells, surface epithelial cells, And we tested for what I mentioned to you. Do they express the mucin, that's mucin 16, and do they express addition molecules such as CD44 or alpha V beta 3? I'll show you here only two. The control, the epithelium, or the surface epithelium of the uterus, express high levels of mucin 16, as you see here with the red color. All the epithelial cells are mucin 16 positive. Classical surface epithelium on a mucosal tissue. And they don't express CD44, one of the addition molecules. When we expose these same cells to factors secreted by the dendritic cells, the mucin disappears. There is a degradation of the mucin and a suppression of the expression of mucin, and there is the induction of CD44 in the surface of the cells. In other words, the inflammatory process necessary for the preparation of the endometrium is led by the dendritic cells, who are secreting factors that act in the epithelium by decreasing the expression of the mucin, releasing or exposing addition molecules that then will lead to the first interaction, the real first maternal, the mother with the baby. This is the first kiss that I like to call between the mother and the baby. The mother now has nothing covering her cells, is having integrins in the surface, and they form these synapses with CD44 and integrins. Also, what we show is there is a lot of expression of osteopontin, and osteopontin function as a bridge between the two sides, and this close this first interaction. So, in conclusion, what I have shown you very quickly is that the process of blastocyst attachment to the epithelium depends on an inflammatory process that we think is mainly led by dendritic cells as well as other cells, but the dendritic cells are critical and they are responsible for removing the mucins and allowing the exposure of addition molecules. So the process of attachment of the blastocyst to the epithelium of the uterus is an inflammatory process and requires this type of cells. So I have a lot of many discussions about this idea, this process with colleagues. And I remember presenting once this lecture, and one of my colleagues in the audience asked me, okay, is this something that is unique to the human? Or is this something that is maintained in the species? How do I know that this is really something real in nature, does exist in other species? So I approached my friend at Yale, Gunther Wagner, who is an evolutionary scientist. He studies the evolution of reproduction. And he has been for many years trying to understand what are the characteristics of the evolutionary steps between the early process of reproduction and the mammalian reproduction. So he studies these 3 stages. He compared the monotremes, who are unique because they lay eggs. They instead rather being live youngs, so they will born from the eggs. However, the monotremes are mammals, although they put eggs outside. What it makes them mammals is because they nurse the young with milk. So in other words, they don't have the classical mammalian pregnancy that is inside of the body of the mother. So when we go to the more closed and evolutionary stages, that is the viviparity, that is part of that the pregnancy takes inside of the body of the mother. We have two subdivisions. One is the marsupials, and second is the eutherians. So what is the difference between the two? That both of them has implantation. The difference with the marsupials is that the implantation is for a very short period, and then the rest of the pregnancy is going to be carried on within the mother, but in a pouch, and there is no placenta. And then we have the eutherians, which was called formerly placentals, and they include the mammals as well as the humans within those mammals, where there is an organ interfacing between the fetus and the mother, that is the placenta. All the mammals who are in part of the eutherians will have an implantation, a placentation, and the fetus will grow within the mother and is separated between this barrier that is the placenta. So Dr. Gunther was studying what are the similarities between the marsupials, and in this case, he used the opossum, and of course, the full mammals who had the placentation. So the opossum, as you can see, has the babies. The babies grow in the punch, similar as the kangaroos. So it has this pouch where they grow. So he was looking at the implantation time, and here you have the egg of the marsupial, the fecundary egg, sorry. This is the uterus of the opossum. This is the surface epithelium of the lumen. This is the stroma. And this is the trophoectoderm. And you can see that the trophoectoderm is interacting with the epithelium. You can see the trophoectoderm and the epithelium. There is a nice connections between the trophoectoderm and the epithelium and the marsupial. But this interaction happens only for 24 hours. And after those 24 hours, there is now a rejection, a separation, as you can see here. It's a reaction in the epithelium. Now is a separation between the embryo, the trophoblast, and the surface epithelium. And this embryo will fall out like an implantation failure. The only difference, see, all these very similar to the humans. The only difference is that the opossum, they have a plan B. So when this embryo did not implant, it goes to the pouch. And in the pouch, the rest of the pregnancy will take care, and there will be a successful pregnancy. In humans, as you know very well, there is not a pouch. So if this implantation doesn't happen, that we will call implantation failure. Now, here comes the interesting thing. Because when he analyzed he did RNA-seq and analyzed all the genes who are expressed in the earliest stages of the implantation, the surprise was that all of them are inflammatory cytokines. And that's when he calls me, and I was delighted to see the data, because the arrow indicates the time of implantation, as you see here. So the arrow is the time of implantation, and at the time, the dominant genes are inflammatory. And that comes a surprise, and when we started the discussion, because he didn't know how to answer that, because the inflammation did not stop at the time of the implantation. The inflammation continues, and it was higher and higher in subsequent days. And the subsequent days, when this inflammation becomes huge, is associated with the separation, or what we call the parturition of the embryo, in order to then go to the pouch. The interesting thing is that these inflammatory cytokines, who we found in the opossum, So interleukin-1 alpha and beta, IL-6, PG, prostaglandins, 6-CSL8. They are present in the sheep, in the pig, in the horse, in the dog, the mouse, and so on. So evolutionary, when there is an implantation process, all those species have inflammation, confirming that inflammation is an evolutionary process necessary for implantation. But then I still have a question. A leaf, for example, is not in the opossum. Important. Remember that leaf is not in the opossum, but is in other species like the sheep, the pig, horse, and so on. But I was telling you, if we go back here, we have an inflammation at the beginning, and the inflammation just continues going up. Why? What is the meaning of that? So let's put it in this graphic, and I will go in more detail. So the opossum have a pre-attachment phase. Then it has an attachment that is associated with inflammation. And that inflammation continues going up. And that is the parturition. And that's when I sent him our definition of pregnancy. So he matched it to the opossum. So we know, as I mentioned to you yesterday, and I summarized quickly today, there is a pre-attachment phase. There is an attachment of inflammation in humans, because it's very similar to the attachment inflammation in the opossum. The difference between the attachment between the opossum and the human is this phase. There is no in the opossum an anti-inflammatory phase. In the opossum, it jumps immediately. In the opossum, it jumps immediately from this one into this one. And we know that this inflammatory step is associated with parturition. So the opossum goes for inflammation for implantation directly to inflammation to parturition. There is no the critical step that we need in humans, that is the stage of placentation. Oh, let me close this. So again, this is the reason we are defining the human pregnancy now, even in the stages that I mentioned to you. We now know that before the pregnancy or the pre-implantation period is anti-inflammatory. The implantation period is pro-inflammatory, and then they start bringing back. In evolution, the critical step between the opossum or the kangaroo or the marsupials and the full placental mammals is this, when there was the shift from pro-inflammatory condition into the anti-inflammatory condition. This and only this stage of anti-inflammation is what allows the formation and the growth of the placenta, a critical evolutionary stage for what we are today. Again, as I mentioned, this is what it makes the mammalian or the placentolous species in development, the first trimester and second trimester. So we look also, and this is interesting because it is a gradual process. It's not that you just shut once the inflammation and then you have the anti-inflammation. No, it's a gradual process, and there is ratios of inflammatory cytokines and anti-inflammatory changes. And that is what it makes it very difficult to do clinical studies, because if you look just at one point, and if you look only one cytokine, thinking, okay, I will go to see only TNF-alpha, you will not get the full picture. It's a gradual process that is taking place during the changes. So we did a study looking at the cytokines starting at day 4 after the embryo transfer and going all the way up to day 15. And if you look at IL-10, You see IL-10 maintains its levels during those days. I will talk about the losers in a moment. And you look at TNF-alpha also. TNF-alpha starts higher and start going down. What it gives us the big picture is when we look at the ratio. If we look at the ratio of IL-10 and TNF-alpha, we see that the ratio is low at the beginning, means there is an inflammatory process at the beginning. Around 6 to 8 weeks, that inflammatory process starts decreasing. When we reach 9 to 3 weeks, that inflammation is gone. However, look what happened in cases of pregnancy loss when we look also at the ratio. They start with an inflammation. Pregnancy loss, sorry, and there was an implantation process. So the loss came later, by the way. There was a miscarriage occurring around week 8 to 12 in these patients. That you see the numbers of patients are decreasing. So we have a successful and a high number who have a clinical as well as biochemical implantation between week 4 to 5. And there was, in all of them, an inflammatory process. As in these patients, the inflammation continues being high. You see the numbers of patients with successful pregnancy start decreasing. And when we go 9 and 3, there is very few who are still holding there. They also lost the babies in after week 15. But the majority of these patients lost the babies because of this continuous inflammation between weeks 6, 8, and 9. So now we think this period between the week 6 to 9 is a critical stage in the shift or the decrease in the ratio between anti and pro-inflammatory. If this shift doesn't happen, the probabilities of losing the baby increase significantly, and it will lead to the miscarriage. This shift, we think, is the critical evolutionary stage that would allow a successful placentation, which will then to the next stages of the fetal growth, at least until reaching the second trimester. So clearly, as you see in this arrow, this process, this change from the anti-inflammatory, what we see is happening in the early stages, is extremely important from evolution, but now for the success of the pregnancy. And again, we can look now in a different way to show the shifts. The normal is the green, doing the ratios. You can see that it's a nice range where this pro-inflammation, anti-inflammation goes. So you start in pro-inflammatory and they start shifting against anti-inflammatory. And you maintain the ratio. Yeah? Sorry, this is the concentration TNF-alpha and IL-10. Let me go, because of time, directly to the ratio. Let me see if I have here. Oh yeah, I was saying. So what the questions now is, of course, what is maintaining this? What are the factors who will lead to the changes on these cytokines that maintains the pregnancy? So here you have a better picture of what is the difference between a healthy, successful implantation and placentation in the first 10 weeks of the pregnancy. So you start with inflammation around week 7, 8, 9, you start now shifting into anti-inflammatory, who remains an anti-inflammatory. In the cases of pregnancy losses, you have a successful implantation, you are associated with inflammation, no difference from the control. However, this is the time where you start looking at the difference. Here, the normal, here at this in this period, the normal, the anti-inflammatory signals now start growing. In the cases we're going to lose the baby, the inflammation continues high. And that continuous inflammation will lead at the end to the loss of the pregnancy, the loss of the baby. So to summarize this part, we have an anti-inflammatory who goes to first inflammation during early pregnancy. This change critical. This is the, as I repeat many times, the evolutionary stage that is what is with today. However, if that evolutionary stage doesn't happen in humans, they will have, if it has continuous inflammation, now they become like the opossum. Yeah. Implantation, detachment, and the opossum goes to the punch. In the humans, they lose the pregnancy. So I have shown you already this first part, how the dendritic cells are changing the process of the epithelium. The next part, the next 20 minutes, what I'm going to discuss with you is, are the inflammation necessary for the second part of the implantation? Because I mentioned to you, attachment is not enough. We need now the trophoblast to migrate and then invade. Yes. And the next question also is what I mentioned, what are the factors that are regulating the shift in each of these stages? Because again, if inflammation is happening too early, as I will show you later, that will induce complete embolization. So what regulates those shifts? So the first question that we have was, what is the source of inflammatory signals? And you will think that the source of inflammatory signals will be immune cells. When we look at the wound, remember the study that I mentioned to you at the beginning, every time that we were wounded, we do a wound, we found multiple cytokines, inflammatory cytokines induced. But one of the inflammatory cytokines that we saw increase after the wound was alpha. TNF alpha increased significantly in the endometrium. Yeah. And as you know very well, and you hear also from Dr. Joaquin yesterday, TNF alpha is one of those cytokines that you find at the time of implantation, and it plays a role regulating NK cell differentiation and so on. So we try to understand what is the role of TNF alpha at the time of implantation, and what are the target cells of TNF alpha that will enhance that inflammatory process. And you see here that we have an arrow going to the dendritic cells, to macrophages, because that's what we originally thought, that maybe TNF alpha is active in the immune cells. We took endometrial cells and we treat them with TNF alpha to see which ones are the ones who respond to TNF alpha. We did also macrophages and so on. For our surprise, that the only cells who have an incredible response to TNF alpha were the endometrial stroma cells. And the endometrial stroma cells, they were producing a lot of additional pro-inflammatory cytokines, but also a lot of the chemokines that were discussed also yesterday, such as GM-CSF and CSF, GRO-alpha, IL-6, MCP1, and so on. So TNF-alpha only in the stroma cells was able to enhance the expression of the production of these chemokines. Yeah? So we ask ourselves whether we hypothesize that maybe the immune cells present at the implantation site, and now we know a lot more about that, especially macrophages. So let me remind you, macrophages at the implantation site, and I think I will talk in another class, the macrophages at the implantation site belongs to the M1 type. So they are pro-inflammatory at the time of implantation. And what we find in us that these macrophages express a lot of TNF alpha. So we question ourselves whether this TNF alpha expressed by macrophages are acting on the stroma cells, and the stroma cells are producing the factors who are directing the trophoctoderm to the migration. So we tested that. We took conditioned media from TNF alpha treated stroma cells, and we put our blastocyst-like structure in this conditioned media. And as you can see in this video, these blastocysts, they attach to the plate, and in a nicely regulated way, they start migrating outside of the blastocyst, and they're covering the whole plate. Let me show you back again. So here is this blastocyst-like structure. It's measured along the trophoectoderms. In 2 to 4 hours, they have attached. And look at that. All the trophoblasts are coming out. Yeah, and they're coming out in a very nicely organized way. We have now ways even how to quantify the rate of migration and the time of migration. Yeah. This is happening when these blastocyst-like structures are exposed to trophoblast stroma conditioned media, TNF-alpha treated stroma conditioned media. Is this specific for the stroma? We take conditioned media from epithelial cells, yeah, or from macrophages, or from B cells or T cells. We try all the immune cells, the factors from all the immune cells. As you can see on the right, this represents the epithelial cells, or B cells, or T cells. In the conditioned media from any of those cells, the blastocyst is all the time floating. It never attached, it never migrate. So what I'm showing you is that only the factors secreted by the stroma cells, and especially stroma cells treated with TNF-alpha, are able to induce this beautiful process of migration of the trophoblast, which then creates an incredible monolayer. I'll show you a little more. Nothing from other cells will do the same effect. So what we are seeing now is that TNF-alpha binds to TNF-alpha receptor and then is inducing the migration. So we wanted to characterize and really define if this circuit. We started with TNF-alpha. So we're knocking down the TNF-alpha receptor. And then we wanted to see if we collect conditioned media from TNF-alpha TNF-alpha receptor, knock down stroma cells, if it still can call the blastocyst. So we took that conditioned media, we put it again into the blastocyst-like structure, and as you can see here, they have some cells coming out, but the process of attachment and the nice migratory process is completely gone, suggesting that again. So that was number one. We took also from the opossum, the conditioned media from the opossum. Remember, in the opossum, the first part was attachment, but there was no migration and invasion. So we thought it's really something missing in the opossum and the stroma of the opossum. And we collected stroma cells from the opossum, we put it in the blastocyst, and a completely different story. Even 48 hours, 72 hours, it hasn't attached, so there is a weak attachment, but there is no migration. So here comes the major evolutionary stage. The stroma, and the stroma who in a way is inflamed, is responsible for telling the trophoblast cells to start migrating. It doesn't have an effect on the epithelium. The epithelium is the dendritic cells. The stroma is a critical component to tell the trophoblast cells to start migrating. So a stroma-derived factors promotes trophoblast migration, and there's only the human stroma cells and human stroma cells exposed to TNF-alpha. So I mentioned to you, this is the migration. What about the invasion? So in order to understand the invasion, we created this model. So we have the trophoblast spheroids, we have matrix gel, and at the bottom we have human stroma cells. We can also replace the human endometrial stroma cells with any type of cells. And the idea is that you have stroma cells, you have this layer of extracellular matrix, and then if the trophoblast really is getting the right signals, after migration should penetrate the extracellular matrix in order to embed within the uterus. And again, it's very similar to the components of the human endometrium. So let me show you some of the data. If we take these blastocyst-like structures having trophoblast cells, and we put them on top of the matrigel, they remain circular, no cells will come out, nothing will move. You can keep them for a long time, even a month. They will look exactly as I show you here. However, if we put human endometrial cells, and they have a big separation between this layer of matrigel. So having the stroma cells in the bottom, the matrigel and the blastocyst, this is at the beginning, day 1. Day 4, you can see already that the shape of these blastocysts are changing. There are projections coming on from the blastocyst-like structure inside of the matrigel. You can see in another picture looking closer to the invasion. Look at the projections. They are within the matrigel. Let me summarize this in time. This is 2 days. This is 4 days. You see the shape of change, the change of shape. There is invasion. There is now penetration within the matrigel. And in 6 days, now there is a network of projections coming inside of the matrigel. 8 days, further these networks are invading. 10 days, it's a beautiful connections all over the culture. Here, let me show you, here what you have is stroma cells. Those are stroma cells. So now, these projections, this invasion of the trophoblast is reaching the stroma cells in the bottom of the plate. And if I close this here, I will show you a picture. You can see here it's the penetration of these projections from the trophoblast. That they are reaching now the stroma cells who are in the bottom of the plate, and they will do the connection. And again, this is looking later into closer to the bottom of the plate, a beautiful interaction between the stroma cells and these invading trophoblast cells. This only will happen when you have the stroma cells. If we put epithelial cells in the bottom, they don't move. If we put now stroma cells where the TNF-alpha receptor knockout has been, having a TNF-alpha receptor knockout, so stroma cells without the TNF-alpha receptor, they also don't move. So clearly is a TNF-alpha activating the stroma cells, secreting the factors that are necessary for the invasion. So I finish now with the models of what we have been talking. So the dendritic cells and additional cells play an important role in the epithelium, the early stages, by removing the mucins and allowing the first exposing the addition molecules and allowing the attachment. That is the first phase. The second phase, the migration and early invasion of the trophoblast, is macrophage TNF-alpha stroma cell mediated. The macrophage secrete TNF-alpha. TNF-alpha binds to its receptor. The induced expression of cytokines. We have done a race of all the cytokines that the TNF-alpha induced expression. That is a manuscript coming soon. And those cytokines are reaching through the matrigel into that attached blastocyst, and it tells them to migrate and invade. And that is now when the blastocyst is embedded within the uterus, and the process of placentation then can continue. If TNF-alpha is not expressed there, or if the seedwall cells are not able to respond to the TNF-alpha because they lack the TNF-alpha receptor. Or if there is excessive inflammation here with a different type of cytokines, the embryo can attach, but the embryo will not penetrate. The blastocyst will attach, but it will not penetrate. And there will not be migration implantation. You can have a second type of implantation failure. So what I'm proposing you that implantation failure is not only a problem on the epithelium, it's also a problem. You can have a good epithelium, you can have a good attachment, but if the stroma cells are not properly educated or are not secreting the factors that they should secrete, the second part of implantation, that is the migration, will not take place, and that will lead to the loss of the pregnancy. So inflammation is necessary for the preparation of the epithelium for attachment. Inflammation mediated by TNF-alpha prepare the stroma to communicate with the blastocyst and promote the process of migration and invasion. Lack of inflammatory signals are associated with poor attachment and defective migration. And as I say, in life, everything is timing, my friends. So I repeat so many times ago, implantation, pro-inflammatory. The shift leads to the best and more important evolutionary stage, that is the formation of the placenta and the growth, the intrauterine growth of the mammal, of the embryo, and the fetus. And then at the end, you have inflammation that leads to parturition. If the inflammatory process associated with parturition occurs too early, that is preterm birth. The same process at the wrong time, you get a disease. If you have a good inflammatory process, you will have an implantation. But if that inflammation continues, you will have an early pregnancy loss. And if you don't have an inflammatory process and it's only a continuous anti-inflammatory, you have implantation failure. Now, the implantation failure, I don't know if you can see at the end of this, can also be if you have excessive inflammation. Too much of a good thing is bad. The lack of inflammation or excessive inflammation will lead to the termination of the pregnancy. These temporary changes, the sequential changes, it's what it makes the evolutionary modifications that lead to what we are today. When those evolutionary process or the modifications don't happen, we go back to be like the kangaroo or the possum, and we lose the attachment. But unfortunately, we don't have a plan B, and therefore the pregnancies fail. And with that, I will stop sharing my screen and cancel. Stop sharing, and I will be happy to answer any question if there is any question.
Thank you, Professor. Very nice, very wonderful lectures.
Thank you.
So right now, so Wang Huan, do you have some kind of question? Could you please speak out?
There is some question. Let me see if I can. Sorry.
Okay.
Maybe if you can.
Professor, can you hear me?
Would you mind to read the question, because I was seeing and they disappeared.
Okay, can you hear me, Professor?
Where are the chats? I couldn't hear you.
the information during implantation is so important. How can we copy the inflammation in women who cannot implant embryo successfully, and how? Thank you.
Yeah, thank you for the question. Oh, I see. Okay, I see the questions here. Good.
Oh, thank you.
Yeah. The inflammation in the implantation is important. So why we copy the inflammation in women who cannot successfully implant? Thank you. Wow, that is an outstanding question, and I hope I will have the answer to you. There is a ways that we are trying to develop interventional things in order to provide that inflammation. The most simplistic way for those cases who the uterus was not implanting is this approach of the wound. But again, the wound doesn't work for everybody. The wound works for a specific group because it's triggered to induce inflammation. We did a study, we looked at several groups of women. Some women will have inflammation after the wound, other women with the same type of wound, at least what we thought is the same type of wound, did not have the inflammation. So we don't know why is the difference. We think the best way will be now to start introducing the factors who are necessary for priming the epithelium, for example, for those that the epithelium is not responding. And our work is focusing on identifying what are those main factors. What are the ones responsible of degrading the mucin and inducing the expression of addition molecules. Once we have identified, we can just deliver into the uterine cavity. We started doing some work, for example, putting HCG, or there are some studies using MCGSF, intrauterine, what is called the intrauterine lavage. The good thing is that once we know what is the most effective factor, we can release it within the uterine cavity and potentially reactivate that inflammatory process, or at least prime that epithelium. Unfortunately, at the present time, we don't have clear candidates. We know some of them, we are testing them. It seems it's not one single one. It seems it's a combination of a few, because some remove the mucins and other ones induce the expression of the addition molecules. I perceive in the future the preparation of a cocktail of these cytokines and chemokines who will be delivered within the endometrium and prepare down the endometrium for the embryo transfer. I hope I answered your question.
And thank you for your detailed explanation. Thank you.
Thank you. There is another question. You mentioned that many kinds of immune cells. Why have I removed this? PCs. I hate PCs. Okay. The next question is, how do the different stages of pro-inflammatory and anti-inflammatory change back and forth? Is the inflammatory response of embryos transferred to different strains of mouse models the same? Very good question. One of the candidates that we have identified that are responsible for the shift is macrophages. We just published a paper showing that HCG is able to switch from M1 to M2. And Professor Liao has shown that PD-1L is able also to shift from from M1 to M2. Both the HCG as well as PD-L1 are secreted by the trophoblast. So our hypothesis is, and we have a nice model in a recent publication, that once the communication between the blastocyst and the mother has established, is the blastocyst that has been able to migrate and invade, the next thing is now start the crosstalk with the mother. And the way that it crosstalk to the mother is by modifying the type of macrophages present at the implantation site. So we show very clear that ACG will suppress the expression of pro-inflammatory cytokines. And PD-L1 will shift the M1 macrophages into the M2 macrophages. When that is established, suppression of inflammation by the stroma cells and suppression of inflammation by the macrophages, now you have the perfect family who will be producing anti-inflammatory cytokines. And the nurture and the growth of that embryo will be now assured, as long as there is no additional factors that will interrupt that. So you see, is the whole shift depends on the communication of the two sides. If the implantation, if the attachment and the first migration is not good, then the signals from the embryo will tell, hey, this marriage did not start in a good foot. We are not happy. And maybe the embryo will not send those signals. The macrophages will remain as an M1. The stroma will continue producing inflammatory cytokines such as CCL12, RANTES. They will recruit the wrong immune cells like cytolytic T cells, and they will reject. Both sides play critical roles. Yeah? In how you can transfer these different strains of mouse and mother, from the evolutionary studies, it suggests that it's the same, those changes in any type of mammals who have placentation. Whether they are exactly the same cytokines like LIF, we know that they are very important in mouse, they are not so important in human. The specific cytokines may be different or vary, but the biological concept and the biological principle is transferred from the humans and the mouse. Let me see, Professor Leal, do you see any other question in the chat? I think you are mute. I cannot hear you.
Sorry. So the second student asked, you mentioned that a lot of immune cells are related to implantation, but she wondered whether B cells play a role in this process. What are its specific functions about B cells? Yeah,
thank you. A very important question. So, you know, time is always a limitation when we teach. And the B cells who were thought for many years that they are not even present in the uterus, now we know they are. So the B cells, they do play a critical role, but their role seems to be more related to the regulation of tolerance. So the expression of, they induce the differentiation of T regulatory cells. So these B cells who are present in the endometrium are extremely important for the recruitment and the differentiation of T regulatory cells. I didn't mention anything else about the other cells like the NK cells because you already hear a lot yesterday. So what I can tell you, what I think is that the NK cells are coming at that stage when the implantation has occurred. The NK cells may also be extremely important on maintaining this anti-inflammatory condition. If the anti-inflammatory condition is not maintained, is when the same NK cells now will trigger the rejection. So those markers that Dr. Joaquin was mentioning in terms of the expression of the NK cells who will be supportive may be also markers of this evolutionary shift, yeah? That it goes from the anti-inflammatory into the pro-inflammatory, that goes from the pro-inflammatory into the anti-inflammatory. And is that something that if Dr. Joaquin is still here, we can discuss in the future, maybe to collaborate. I leave you a lot of ideas. Many of these things need still to be processed and tested, but that is the objective of this type of courses, to bring ideas for discussion and for collaborations, as the very productive collaboration we have with Professor Liao, which I hope we will continue doing in spite of all the regulatory and political problems. What else?
Thank you. Yanbei, could you please speak out your question, Yanbei?
Okay. Thank you, Professor. I have learned a lot from it. I would like to ask how dendritic cells affect embryo transfer. Thank you.
Even dendritic cells will affect the embryo transfer. That's your question?
Yes.
Yeah. So the dendritic cells, as you know very well, there is different types, and it depends on the stage of activation. What we see is that the dendritic cells who are present at the implantation site, they are unique. They are tolerogenic dendritic cells, and they are associated with the function of supporting the tissue. Can we use those dendritic cells to prepare the endometrium as cell transfer that is now becoming more and more hot, a very hot subject? I'll tell you the truth. I don't know. I really don't know. The idea is brilliant. I love your idea. I don't have an answer because we have not been able to isolate those dendritic cells. Or let me put it in other words. We have not been able to replicate in vitro the characteristics of the dendritic cells at the time of implantation. Yeah? That has been the challenge. If we will be able to replicate in vitro all the characteristics and then potentially do a cell transfer into the peritoneal cavity, and with the idea that they then will do what they have to do, that will be fantastic. Unfortunately, the barrier that we are dealing with is to be able to replicate that phenotype in vitro. Dendritic cells are very difficult to differentiate, as you know very well. I answer your question?
Thank you.
I think there is another question here.
Yeah, Wang Jing, could you please speak out your question, Wang Jing?
Oh, yes. Can you hear me?
Yes.
Oh, okay. Dear Professor, I want to ask a question. At the end of pregnancy, the mother will also show a poor inflammatory state. Is this state completely consistent with the poor inflammatory state at the term of embryo implantation?
Yeah. Yes. So we know that parturition is a stage of inflammation. So if we look at the cytokines and the factors that were present in the opossum at the time of the detachment, they are prostaglandins, they are high levels of IL-6, interleukin-1 beta, and so on, TNF-alpha. There is M1 macrophages. All those are characteristic of the inflammation that is happening in normal parturition. Yeah. You know very well, prostaglandins are major mediators of parturition. Interleukin-1 beta, IL-6, the influx of macrophages into the muscle to induce the inflammatory process. All those are the characteristics of parturition. The same thing with the same characteristics are happening earlier, and that's what we define preterm birth. What is triggering that inflammation to happen at the wrong time? That is a different question. Or what is preventing the shift from the anti-inflammatory into the pro-inflammatory? That is another question. Yeah? And that is a question for a long discussion that maybe I will show some evidence in the future classes. But the biological components, and your question is very good, are the same. So they are happening. It's just they are happening at the wrong time.
Okay, thank you.
But just to clarify, the pro-inflammatory signals necessary for implantation is not exactly the same necessary for the parturition. And also is the degree of inflammation. The degree of inflammation at the time of implantation is moderate, low. It's inflammatory, but it's well controlled. The inflammation that is happening in the parturition is 10 times higher. Yeah. So the molecules are the same, the concentrations are different.
Okay, thank you very much.
Welcome.
Okay. Next question is Dan Yun Qin. Yeah, could you please speak out your question? Dan Yun Qin. Okay, so
hello, can you hear me?
Yes, we can hear you.
I have a question that what's the relationship between HLA-DR positive and CD11C positive dendritic cells and endometrial stroma cells?
Very good question. We did not see that the dendritic cells will be able to replicate what we see with the macrophages. But again, I have to be clear that the differentiation of the dendritic cells that we are using in vitro may not represent the same characteristics of the dendritic cells that we see in the human endometrium, the normal. And that is a technical limitation that we have. We are trying to understand this question, and your question is very important. At least in our hands, and if somebody can help us, I will be delighted to collaborate. We have not reached the level to replicate the same type of dendritic cells that we see in endometrium in vitro, in the lab, in order to get conditioned media and study them. Isolating also from the endometrium in early pregnancy is a challenge because the percentage of these cells are very few. And in addition, now we have more problems because we don't have access anymore to this tissue. So a project that I had in terms of studying this CD11B, HLA-DR positive, CD11C positive, we had to put it on hold because we don't have access anymore to early tissues to study that. Yeah. What is the characteristics after early implantation? But it's an important question. It's a very important question.
so next question.
Thank you.
Yeah, okay, thank you. So next questions maybe is from Liao Zhiqi. Could you please speak out your question?
Okay, hi there, can you hear me?
Yes.
Oh, hi, Professor Gil. You said that TNF-alpha and TNF receptor in human stem cells are vital for embryo implantation, and we all know that there is a fetal and maternal immune interaction. So I want to know, how do the trophoblast cells affect the human stem cells or the decidual cells? Thank you.
Thank you for the question. Excellent question. And this is what we have been working, because I'm telling you, the stroma is inflamed. There is all expression of this inflammatory cytokine. What will be the shift? And is the stroma cell. The stroma cells, one of the early hormones that the stroma cells produce is, one of the early hormones that the blastocysts produce, the early hormone that the blastocysts produce, that is produced by the trophoectoderm, or later by the placenta, is human chorionic gonadotropin, HCG. HCG binds to a specific promoters of the chemokines who maintain the inflammation, and HCG induce a silencing, they silence the endometrium, the stroma cells. They block completely the inflammation. And this silencing, this inhibition is epigenetic. So it does modifications on the histones, induce histone modifications, and the histone modifications will completely block the production of pro-inflammatory cytokines in the stroma that even TNF-alpha cannot overcome. So even if you still have some TNF-alpha there, when HCG is acting, will completely block the production of pro-inflammatory cytokines in the stroma. Yeah? So the role of the placenta, or the early trophoblast, because still there is not a placenta, the role of the early placentation of the early trophoblast is to silence the stroma cells and their inflammation, and to shift them into a non-inflammatory condition. Good. I think because of time, we will stop. Yeah?
Yeah.
I really want to thank you for all of you and your outstanding questions. I'm delighted. It was a great idea that you write the questions, and thank you for reading to me. And we will see you tomorrow.
Okay. Yeah.
Okay. Thank you. Have a good evening.
Yeah, have a good day.
Bye. Bye.